Hello friends, welcome to the second lecture on cancer diagnosis. In this lecture, we will first give a brief introduction about the different cancer biomarkers and then we will discuss about the nucleic acid based markers in cancer diagnosis. So what is a biomarker? We now know that the biomarkers are nothing but any compound or any substance which can indicate a particular biological state. Biological state means it's an inherent biological stage of an organism. Also, the biomarkers can localize a particular molecule. It can be also a marker. Now, there are two key parameters of a biomarker. These are the sensitivity, which is the measure of the marker's ability to detect true cases of a specific cancer, and the level of sensitivity is the minimum amount of that marker which can be found in the cancer cells. So 98% sensitivity of a marker means that in 98 cases out of 100 tested, a cancer can be detected in a clinical sample with that marker. The next parameter is the specificity, which is the measure of the marker's ability to differentiate or discriminate between patients or between patients with true disease from those which are free of disease or the non-cancer patients. Hence, a specific marker will give negative results. A specific marker or a with high specificity will give negative results in a person free of disease. So, how we can calculate these two parameters? So, sensitivity, as you can see here, it is the number of patients with positive tests divided by the number of patients with the actual disease. So, a sensitivity of a marker defines like that. So, how many patients? How many patients can have these markers out of the total patients which are who are having the diseases? So, if it is found maximum is the number of the sensitivity, higher the level, maximum is the number of the patients with that particular marker, higher the level of sensitivity. On the other hand, specificity can be calculated from the number of patients with actual diseases, which are the true positives, divided by the number of total positive tests. That means a marker can be found in the patients with and without the disease as well. So the persons who are not having the disease but still having the marker, they are the false positives for this disease, whereas the patients who are positive for the marker as well as positive for the disease, they are the true positives. So these are the two parameters which has to be balanced, or you can say it can be optimized for a specific marker like the sensitivity and specificity. Now there can be different types of cancer biomarkers: nucleic acid based, protein based, carbohydrate, lipid based, as well as small metabolite. Whole cell based markers are also developed. Now cancer biomarkers, it can greatly improve the cancer detection, diagnosis and treatment methodologies. A cancer biomarker definitely can reduce the cost of cancer diagnosis and treatment. Now, let us see about the different types or categories of tumor biomarkers. A number of tumor biomarkers have been identified and they are effectively being used as cancer markers. There are nucleic acid based markers like mutations, loss of genetic heterogeneity, uh, microsatellite instability and gene expression, as well as protein markers and protein pattern recognition profiles, circulating tumor cells and circulating endothelial cells. Many of these markers can be detected in the circulation, whereas other biomarkers can be found within the tumor tissue itself. So in the next few slides, we'll discuss about the nucleic acid based biomarkers in cancers. Several genetic modifications can be found within the tumor cells, and some of these are useful in cancer diagnosis and staging. Tumor derived DNA can be detected in plasma, urine, or stool samples of the patient. DNA modifications can be estimated by checking whether there are any loss of heterozygosity, presence of mutations, microsatellite DNA alterations, DNA methylation patterns. But one thing that we need to keep in mind that blood plasma may contain DNA from cancerous as well as from non-cancerous sources as well, like during a heavy infection or autoimmune diseases or self-degradation of the plasma DNA. All these may contribute to a problem in assessing the real cancer cases. Detection of cancer-associated mutations in the body fluids were first observed when fragments of the p53 gene were detected in the urine of bladder cancer patients, and RAS gene mutations were found in the stool samples from patients with colorectal cancers. Other DNA mutations have also been detected in cancer patients' blood, urine, or other body fluids. Loss of heterozygosity. It occurs when one copy of an autosomal gene is lost due to some reason. It is very common mutation in cancers. Specifically, it has been observed that one of the tumor suppressor gene allele is lost where the other allele is mutated. Definitely cancer will develop then. So Knutson's two-hit hypothesis is actually based on this phenomenon where first mutation may shut off one allele whereas the second mutation in the other normal allele will totally result in loss of function. This LOH is a hallmark of many cancers and can be detected by PCR-based methods. Next is the microsatellite DNA markers. These can also be used as a method to detect the loss of heterozygosity. To recall, microsatellites are stretches of repetitive DNA sequences which are about 6 base pair long and can be repeated 5 to 50 times. Thousands of such repeats can be found in the entire genomic length of the organism. So these number of microsatellites are altered in cancer cells. The microsatellite DNA instability can be a useful marker which can be detected in the urines of bladder cancer patients. Also, altered microsatellite patterns have been found in the saliva of head and neck cancer patients and in the plasma of patients with small cell lung cancers. Next is the DNA methylation patterns, which frequently changes in the tumor. DNA methylation patterns are developmentally regulated, mostly cell type specific. As we know that methylation of the cytosine residues in the CpG islands in the promoter region of various genes are used to suppress the gene expression, and majority of the genes which follows this kind of regulations are tumor suppressor genes. Hypermethylation can result in loss of function of these genes. So PCR based assays have been developed to accurately detect the DNA methylation patterns in cancer tissue and in body fluids. Such DNA methylation pattern analysis can be used also to distinguish malignant from benign tumors. Mitochondrial DNA dysfunctions, DNA mutations. So mitochondrial dysfunctions are well known in majority of the cancers, and one of the main reasons for mitochondrial defects are the mtDNA mutations. mtDNA is very prone to mutations like that of their nuclear counterpart, but the repair mechanisms for mtDNA is very poor. Hence, the mtDNA mutations are more frequent when exposed to carcinogens. Tumor-associated mutations in mitochondrial DNA have been found in colorectal, lung, head and neck, bladder, and breast cancers. Because mitochondrial DNA is much smaller than the nuclear genome, it is easier to detect mutations in mitochondrial DNA samples. Such alterations can also be found in the body fluid samples. For example, in patients with lung cancer, 200-fold more mitochondrial DNA mutations can be detected in p53 gene mutations. The last one we will talk about is the viral DNA, which can be detected in virus-induced human cancers. For example, human papillomavirus is in the cervical carcinoma samples. Hence, HPV DNA is a good marker for detecting the occurrences of cervical cancers. Epstein-Barr virus DNA in the plasma of patients with nasopharyngeal carcinoma is another example. These markers can be used to monitor response to therapy as well as for the tumor recurrence. With this particular information, we come to an end of this lecture on tumor biomarkers, more specifically nucleic acid based biomarkers. Hope you have enjoyed it. See you in the next lecture of emerging techniques of cancer biomarker discoveries. Thank you.